Everyone, thank you for joining. My name's Ian. I'm a grad student at Princeton in the Metcalf Lab, and I'm also a scientist here at the Rocky Mountain Biological Laboratory. And today I'm going to be sharing a little bit about the Flax project, which is a project about plant infectious diseases that I started just a few years ago. So I'm really excited to share this project. It's sort of in a really exciting stage because in a lot of ways it's still in its infancy. And over the past few weeks, if you've tuned into some of the other lab chats, you've heard about these really cool projects like the Marmot project, the Ground Squirrel project that have been going for decades. But new projects like this are really exciting too, and I'm excited to share all of that with you. Just got to figure out how to actually flip through my slides. There we go. All right, so I just wanted to jump to this figure really quick. For those of you that participated in the science challenge this past week, this will probably look really familiar. Because I'm going to be talking about plant infectious diseases, I wanted to start by just going over what an infectious disease is. So an infectious disease is a disease. I'm sure that makes sense, but it's different from other diseases or non-infectious diseases in that it can spread between organisms. And we use this thing called the disease triangle in a field called disease ecology to understand how an infectious disease actually happens. So it's in an interaction between a host, so that's the organism getting the disease, a pathogen, the organism causing a disease, and the environment in which the host and pathogen are interacting. So this field of disease ecology deals with host-pathogen interactions in an ecological and evolutionary context. So this is basically all about infectious disease in terms of the interactions. So rather than dive into all of the different subdisciplines, I just wanted to give you a quick overview of some things that we think about in the field of disease ecology so you can get a better sense for it. So some of these things have to deal with like where infectious diseases actually come from. So understanding host shifts in zoonotic disease, basically diseases that spill over from animals into humans or vice versa, or between different species of animals. We also try to understand the ecosystem impacts of infectious disease. Here's a picture of a bunch of pine trees that have actually been killed by a beetle. And for those of you in Colorado, I'm sure that you have heard of this problem before. Also, epidemiology falls under the big umbrella of disease ecology. Due to COVID, I'm sure most of you have heard this term and field discussed in the past several months. And epidemiology is all about using mathematics and statistics to understand the spread of disease within a population. So I actually do a lot of my research in human epidemiology when I'm not at Rumble with diseases like COVID or pertussis or whooping cough, using mathematical tools to understand population spread. Actually, what I'm going to be talking about today with Flax is about bringing some of those mathematical tools into the plant world to use them to better understand what's going on with plant infectious diseases. Also in disease ecology, we think a lot about evolution and how hosts and pathogens are co-evolving together in sort of this never-ending battle. So we can use this knowledge to better predict what infectious disease systems are going to do as the environment shifts. So a lot of these topics in disease ecology seem like they apply directly to human systems or animal systems, but plant systems don't necessarily immediately come to mind when you're thinking about pathogens. So here I've just thrown up a few different examples of different plant pathogen systems, and they really span a wide variety of both host systems and sort of applicability to society and things like that. A lot of plant pathogen systems that scientists study, like anther smut or cedar apple rust, might have some implications for agriculture or things like that, but really we study them to understand how pathogens affect ecosystems and ecosystem stability. Because pathogens are ubiquitous organisms, they're really important for understanding how nature works. But it's also really important to study plant pathogens because they affect society, particularly our food supply. So particularly diseases like potato blight or brown spot disease of rice have caused epidemics that have resulted in millions of deaths. So I think it's equally as important to study and understand plant pathogens as it is to study human diseases like COVID or flu or other things like that. So that brings us to the Flax system, which is what I work on here at Rumble. So I wanted to sort of frame this using the disease triangle. So flax rust is a fungal disease that affects flax flowers. So the flax flower that grows here around Rumble is called Lewis flax. If you're in Colorado, I'm sure you've seen this before. It's got these pretty blue flowers and it grows in subalpine to sort of the lower limit of alpine ecosystems. And the pathogen that causes flax rust is called flax rust. And like I said, it's a fungal pathogen. And it infects the stems and the leaves of flax, and it actually transmits within and between flax plants by blowing in the wind. So this orange stuff that you see here on the stem of the infected plant in the middle of the triangle is flax rust. And some of my research assistants dubbed it Cheeto dust earlier this season because that's what it looks like. And it sort of spreads like Cheeto dust. You can think of it as sort of just like, it's almost like a plant sneeze. When the wind blows, the stuff just gets everywhere. And I'll talk about a little bit more about how we measure that later. But flax rust disease is really a product between the host pathogen and the environment. I'll talk a lot about the environment later, but the environment in which this disease occurs ranges from sort of drier, lower elevations to higher elevations. We've actually observed it above 11,000 feet. So it really spans a wide variety of different temperature and humidity conditions. And that's actually one of the reasons why we find it so interesting. Because it occurs in all these different conditions, we can use those conditions to understand how changes in the environment, such as those due to global warming and global change, might affect plant pathogen systems more generally. And you might wonder, why do we study this alpine wildflower to understand plant pathogen systems? Well, believe it or not, this system isn't as removed from agricultural systems as you might think. Lewis flax is actually a close relative of agriculturally grown flax, so flax that you use to get flax seeds or linen and things like that. So understanding what's going on in this system has some important implications for agriculture and things like that. But more than that, flax rust is a good model system for plant pathogens more generally. It's used as a model for understanding how the plant immune system affects disease spread and many other things. So we think of it more as like a lab rat than a system that we have specific questions about. Although the system in and of itself is very interesting and we're learning a lot from it. So before I get into some of the work that we actually do in the flax rust system, I just wanted to quickly introduce you to the flax rust team. So from left to right in this picture, we have myself, and then Maria, Sarah, Barrett, Juliana, and McKenna. And actually, a lot of them are here today to help answer some of the questions that you might have after I run through my slides. So we use the flax rust system to go after a few different big questions about plant pathogens. So the one I'm going to be talking about today has to deal with how climate change might affect the way that plant pathogens spread in space and time. But we also use this system to try to understand what actually limits the evolution of plant pathogens and how climate change might shift those limits. So here's a little roadmap of the project or idea that I'm going to talk to you about today. And that idea, of course, like I said before, is how climate change might affect the way that plant pathogens spread in space and time. So I'm going to start by talking about how we measure environmental conditions and how we simulate climate change using elevation. Then I'm going to talk about how that relates to the within host spread of disease, how we use our knowledge of the within host spread of disease to actually measure transmission, and then how we actually connect transmission to the population level epidemiology of flax rust. And here, I just want to point out in these sort of cartoons that are going to be popping up again throughout the talk, the yellow flowers represent diseased plants or diseased stems, and these yellow circles represent sort of the cloud of spores that infected plants spew out into space and onto other plants, potentially infecting them. So we're going to connect all of these ideas and then actually talk about how we combine them into a mathematical computer model that we can use to actually make predictions about how climate change might change the way that plant pathogens spread. So to get started, I'm going to talk about environmental conditions. So here are pictures of our 4 study sites in the vicinity of Rumble here in Gothic. So our lowest is Cement Creek. For those of you familiar with the area, this is by the Caves Trail and Crested Butte South. And we have sites going up in elevation by the bus turnaround right outside of Gothic, on the slopes of Gothic Mountain, and at a high meadow right before the edge of the tree line on Gothic Mountain up above 11,000 feet. And we chose these 4 sites because they span a wide range of environmental conditions. Generally lower, it's hotter during the day. Cement Creek especially is pretty dry. And as you go up, the conditions change. And this sort of elevational gradient serves as a very rough proxy for climate change, where lower elevations are hotter, and as you go up, things get cooler. So we measure the different environmental conditions like wind speed and direction, temperature, rainfall, humidity, solar radiation, and leaf wetness, soil moisture, as well as a few other things at each of these sites using these weather stations. And then with that data, we can finally start to get into how those environmental conditions affect the spread of flax rust. So we start at the scale of a single point of infection. Here in this picture of a leaf, you can see these little orange circles. Those are called pustules, and that's basically a tiny little colony of fungus growing on the leaf of a flax plant. What we do is we go back to individual leaves on specific plants at each site every single week, and we take a picture of those leaves with some sort of scale or ruler in the picture. That allows us to actually measure the change in the size of these pustules week after week to get at how environmental conditions affect the core biological trait of the pathogen, which is its growth rate. Then once we understand that relationship, we can scale it up to how the growth rate affects the spread of disease within a single stem, and then also how the spread of disease within a stem connects to the spread of disease between stems within a single plant. We have some preliminary results from our work last year that indicate that warm temperature and low humidity appear to speed up the within host spread of disease. But we're really excited to get more detailed data this year and work out these relationships a little bit better. To give you an idea of what this work actually looks like, I put in this picture here. And essentially, we spend a lot of time bending over, putting our faces really close to flax plants to try to find specific leaves and make these tiny little detailed measurements. And we do this for hundreds of plants across our 4 sites. So the next sort of scale of investigation is transmission. So how disease actually spreads away from a single infected plant. So we know how environmental conditions affect the intensity of infection and the spread of disease within a single plant, which is what I showed on the previous slide. And now we want to connect that to the disease dispersing from an infected plant to healthy plants. So the way that we do that is we put these spore traps out in the field around infected plants. These are the tiny little red squares that you can see in this picture. And on those red squares, we put pieces of tape, and we leave those out in the field for about a week, and we in arrays around infected plants. Then we collect them and we can take them back to the lab and actually count the number of spores that landed on each spore trap. And then using all of that data, we can actually infer how many spores are going out from an infected plant in which distances and in which directions. So that's sort of represented by this cartoon on the right with this yellow cloud of spores, or what we call a dispersal kernel around an infected plant. So finally, we want to link these spatial patterns of transmission to the spread of disease within a population. So what we do is we go to each of our 4 sites, and every single week, we find and locate each newly infected plant. We actually lay all of our sites out on a grid to facilitate this. What you can see in this picture on the right is what it looks like in practice. When we find a newly infected plant, we stick in a flag right next to it and record its location. So over the course of a season, the flags sort of let you see how the epidemic grows and eventually sort of explodes. The site on the right, or the site picture on the right, is our bus turnaround site, which corresponds to the red line in this figure. So this figure is showing disease prevalence over time last season. So what we observed is that pretty much every plant at the bus turnaround site became infected by the end of the season, thanks to this massive explosion in disease that happened in the middle of the season. However, at some of our other sites, we saw a very different pattern. Disease definitely spread, but it didn't really take off like it did at the bus turnaround. So we think that a lot of this might be due to the environmental conditions that are affecting the within host spread of the disease and scaling up to different patterns in transmission. But we think a lot of this might also be due to some details of the plant immune system that I don't have time to talk about today. But I just wanted to highlight that we're also using flax rust to really study how the plant immune system modulates the spread of infectious disease, just like the human immune system modulates the spread of things like flu or coronavirus. So now that we have all of these different data streams, we need to put them together to actually make predictions about what climate change might do to plant pathogen epidemics. So I'm actually going to turn things over to Juliana, one of my research assistants. She actually did this for her senior thesis last year. She's going to tell you a little bit more about how all of this works.
Hi, everyone. So basically, we created a computer model, as Ian said, and the basic framework for this model was that environmental processes, such as the temperature that it is and the humidity, can affect how the disease spreads within an individual host. And as the disease spreads more within an individual host, it can disperse more spores onto neighboring plants. And then healthy plants become infected as a result of getting those spores. And the process iterates and ultimately spreads across a large segment of the population. And so using this model, we calibrated it using our data that we collected last year and used the relationships that we found between temperature and humidity to predict how climate change and global warming might alter the spread of disease within the system. And as you can see with the figure on the right, we expect using our preliminary results, and we weren't able to do all of our research because of COVID and lab interruptions, but we suspect that under warming temperatures, the disease will spread more quickly across the population. And Ian will take it home.
Yeah, so this sort of illustrates why we use the Flax rust system to understand all of this. Because we're able to go into the field and measure all of these tiny little parts and fit them together, we actually have a really powerful tool for understanding how climate change might predict or might affect the way that plant infectious diseases spread. And we hope by that using Flax rust as a model, we'll actually be able to make some inferences that are going to carry over into agriculture and other systems, and really help us better understand what's going on with plant pathogens as our world warms up. So with that, I'd like to just thank my funding sources and my advisor, Jess, and I'd be happy to answer some questions and chat more about any of this. So let me figure out how to get back to Zoom. I'll stop sharing. All right.
So everyone, feel free to type into the chat box if you have questions, or just to unmute yourself. And I actually have a question, so maybe I'll start us off, Ian. I'm curious, in choosing your sites, did you look for a certain percentage of existing infection already, or how did you choose that? So you talked about how you chose it across elevations, but what about infection already present?
Yeah, so that's an interesting question, because when I started working on this system, it was sort of an accident, actually. I actually came to Rumble for the first time to work on a different plant pathogen. And when I came to Rumble, it was a really, really dry year. And that actually made it so that the fungus I was looking for couldn't grow at all. So I didn't find any of the disease plants I was looking for. But then I think it was at Cement Creek, I was just walking up the caves trail and I saw a flax plant with this orange stuff all over it. And I had no idea what it was. And I emailed a friend and asked if she knew what it was. And she said, oh yeah, that's flax rust. And as I was trying to figure out how to sort of salvage my field season that had been wrecked by the total lack of disease, I just figured that I should start tracking the spread of disease at that site. So that site was sort of chosen by accident, and the same for the bus turnaround. It was just one of the first places where I saw disease. But as my ideas about putting all of these different scales of transmission together and linking them to environmental conditions to understand plant pathogens and climate change, like as all of those ideas came together, I specifically sought out some higher elevation sites like the Gothic Mountain site and the High Meadow site to sort of capture the extreme end of environmental conditions. So I can put all of the different sites which have, when you go out and see them, and you can see this a little bit from the pictures, they all have very different patterns of temperature, humidity, and everything like that. Because I capture such a big range, it helps bring all of the ideas together, because I just capture a wide range of different environmental conditions across the sites.
Mm-hmm. I have another question.
Mhm.
I'm curious. So originally you were actually studying disease in humans, and then you've switched to plants. What caused the original interest and why the switch?
So I haven't actually switched. So I'm doing a lot of research on coronavirus right now with a group at Princeton. And I do still have some active projects with other human infectious diseases. But I sort of got my start about infectious disease, like most people do, thinking about human pathogens. So when I was an undergrad at the University of Virginia, I somehow, well, it was actually in a course taught by Yanis Antanovics, who later became my mentor. But he talked about using mathematical models to understand the spread of disease. And I just thought it was really cool. So I initially started working with him because I wanted to build, actually, it's such a weird time right now. I'm sure a lot of people have heard the term SIR model in the news, but I wanted to build these sort of mathematical models of disease spread for flu. And I sort of got started with that, messed around a little bit. And then one day he asked me if I wanted to go study plant pathogens. And I thought that was crazy. And I didn't understand why anyone would want to do that. But once I actually got out to the field and got some hands-on experience with that, and just started thinking about the problem a little bit more deeply, I realized that like those are just as important as human diseases because they affect us in many of the same ways. Well, indirectly, of course, by affecting our food and the environment and things like that. And then when I started grad school, I sort of had this weird idea of doing human and plant diseases at the same time. And I was lucky to find an advisor who knew how to do both and was open to the crazy idea of me working in the mountains during the summer and then going back to Princeton and building computer models for the rest of the year. But it's definitely an odd combination, but there are actually a handful of people in the infectious disease world that do both. And I think that by combining ideas from the human animal world with ideas from the plant world, you can actually generate a lot of insights into how infectious diseases work and spread, which is really important as our world is changing and sort of a lot of our paradigms about how we think about pathogen evolution and spread are broken a little bit as things change. Bringing and like cross-pollinating ideas from the two different disciplines, I think, is really important.
Mm-hmm.
Looks like
Right.
Malcolm.
Hey, Ian, I have a question.
Yeah.
Can
Mhm.
you hear me all right?
Yeah.
Awesome. So I saw that on most of your charts, your time axis was in weeks as units.
Mhm.
And in some of those, the slope looked quite sharp. So I was wondering how much the granularity of time matters when you're taking measurements. Or, you know, is weeks good enough? I saw some of them were between a week in relation to, you know, how fast the disease is spreading.
Yeah, that's a great question. So time definitely matters a lot, but it matters in different amounts at different scales. So one thing that we're actually doing different this year is we're taking those pictures of the pustules with the ruler in the picture multiple times per week at each site. And this is because some of what we saw last year, where things just changed so much between weeks that when we're doing the statistics, things got a little bit messy and it would just be better to have finer grained data there. We're still, for the most part, doing the epidemiological surveys where we find the newly infected plants just once a week. That's partly because that just takes a really long time. But also when we're thinking about disease spread, we know that it probably happens on the scale of hours to days, but given our limited time and resources, getting a snapshot every week is actually pretty good. And it's funny because now with coronavirus and testing, everyone is very aware of these data streams and getting updates on prevalence every single day. But for a lot of disease systems out there, you might get prevalence measurements once a season. And those measurements might be very messy and hard to interpret. And in many ways, by going to each site about once each week, we're actually getting an extremely detailed data set of prevalence over time. Because it's also spatially located, we can do a lot with it to understand the spread of disease. But of course, in an ideal world, I could have a team of 100 people out measuring the change in prevalence at each site every day. But I don't know if that would be the best use of resources. It seems like from the modeling and statistics that we've done so far, we're able to make pretty good inferences with our 1 week resolution.
We have another question from Karen. It says, would love to hear more about the arms race between the Cheeto dust and the flax plants. Some defenses both parties have.
That's an excellent question. So by arms race, I think Karen's talking about coevolution. And usually colloquially in science, we do call it an arms race because that's really what it is. It's the host trying to evolve to be able to resist infection by the pathogen, and the pathogen trying to evolve to be able to infect the host. And it's sort of this evolutionary game of cat and mouse that sort of runs on indefinitely. So on the host side of things, hosts try to evolve resistance to the pathogens. And in plant pathogens, I'll try not to dive too deep into plant immunology right now, but plant immunity is governed by what we call resistance genes. So in sort of a cartoonish way of thinking about it, if the plant has a resistance gene that can recognize some part of the pathogen, it's able to mount an immune response and defend itself against infection. So on the pathogen side of the game, pathogens are trying to continue to be able to infect the host or to gain the ability to infect a host they previously weren't able to infect. And when I say trying, that's anthropomorphizing things. These are all evolutionary pressures, not conscious actions on the parts of either plants or plant pathogens. But anyways, because the host is trying to recognize the pathogen to mount an immune response, the pathogen is actually trying to hide. So pathogens can retain their ability or gain the ability to infect hosts by sort of getting rid of the proteins that the plant is trying to infect, that the plant is trying to detect on the pathogen. So this can lead to sort of cycling of genotypes or alleles within host and pathogen populations, and it gives rise to some really interesting evolutionary dynamics. And this is actually part of the reason why I chose to work in the Flax rest system, because I want to link those sort of evolutionary dynamics to some of the evolutionary dynamics that people normally associate with human and animal systems, which are all about why pathogens don't do more harm to hosts and things like that. And there's some interesting evolutionary trade-offs that I'm hoping to explore as soon as I'm able to get back to the lab and start growing up some plants in a greenhouse and doing inoculation studies. But this system has actually been used for a really long time to study coevolution between plants and pathogens. And oddly enough, it was the first system in which the plant immune system was really described in a meaningful way. So I really do think it might not be the system that jumps into everyone's head when they think about plant pathogens, but I think it really has the potential to sort of be, like I said before, like the lab rat of plant pathogen disease ecology and evolution.
I have another question, kind of related to that, but just in thinking about resilience. So in this time with focus on the spread of COVID, or maybe in your case today, both COVID and flax rest, can you give any examples from your career of resilience strategies that you've seen that you have thought were really fascinating or even just encouraging?
Sure, I might actually turn this one over to some of my research assistants. Do any of y'all have an answer you'd like to share? I can take this one too, so I don't have to dump this somewhat tricky question on you.
Well, I could talk.
Yeah.
Well, I'm really backlit right here, but so you might have noticed in the graphs that some of the populations didn't get as diseased as the other populations. So there was prevalence, which was hovering around 30%. And we think that this has to do with plant resistance because it wasn't, because the disease curves kind of followed a similar trajectory. So, and there wasn't really a clear environmental driver of this that we could detect from our model. So these plants are clearly evolving resistance to this pathogen, and it has interesting implications for human agricultural systems because it's typically not the way that we engineer resistance in crops. We typically do large monocultures, which are really very, very vulnerable to disease invasions. So, so there's different, ways that nature engineers resilience, which might be useful for improving our agricultural systems and making them more robust to changes in climate and pathogen invasions.
So that was extremely well said.
Other questions for Ian or his team?
I have another one.
Yeah.
So Ian, I know that you've researched many different places from the Bahamas to the Alps. And I was wondering if you could share how this experience of isolation due to the virus has sort of changed the way you do research. Does the isolation make it easier somehow? Does it really not affect the scientific process?
Yeah, that's a really good question. Rumble has done a truly excellent job of making sure that scientists can continue their important fieldwork during the course of this pandemic, but it's definitely different this year. In some ways, I do think it's easier just because it's sad that there are less talks, there are fewer or actually no real like social gatherings or anything like that. And that does create more time for focusing on research. But a lot of how you actually do research, especially at a place like Rumble, really involves talking to people who study things that are different than what you study and hearing their ideas and having their perspectives sort of change the way that you think about things. But it's definitely different. Actually, I might turn it over to my students again too, because I know that the student experience and the research assistant experience is totally different this year. Actually, one of my research assistants came to Rumble before I even did, so she might have some interesting perspectives on this as well.
Yeah, we're definitely lucky to be able to come here and be able to do research. I know like a lot of places you're not able to. Like last summer I was actually in Madagascar, but this organization that was based out of the UK doing my senior thesis there, and I know like they weren't able to go there at all, and like this is the first time in like 10 years that like no scientists are doing research there. So I think it definitely depends on where you are. I think other countries, obviously it's really difficult to travel there to do research, but fortunately, because yeah, Rumble's still in the US, we're able to come here. And in terms of, yeah, this year it is definitely different. It feels emptier and there's like less people walking around just because there like are way fewer people here. I know a lot of like the REU students weren't able to come. So it's definitely, yeah, less people. We kind of have to keep to ourselves, which is a little bit different, and don't get interact with other scientists and students, which is a little bit sad, but I'm still thankful that we were able to make it here. Anyone else? Give it back to Ian.
All right. Well, Ian, thank you for your time sharing about your research with us today. We really appreciate it. And do we have any final questions before we all sign off? Any closing comments from you, Ian? Final statements you want us to all think about as we go into this world full of pathogens and infectious diseases?
Well, I mean, I'd definitely like to thank everyone for tuning in. It's so exciting to be able to share this project with you. And I really appreciate you taking the time to listen and learn and everything like that. As far as closing statements about pathogens and how they affect the world and everything like that, well, it's definitely an interesting time to be thinking about pathogens. I'm part of a small group of people who thinks about pathogens all the time, and it's definitely strange for the entire world to be focused on things like coronavirus. But I think it's important to remember that pathogens have always been major players in our ecosystems, and they do a lot to actually promote ecosystem stability and things like that. Of course, we need to do a better job of understanding the risk to society and like humanity in general of pathogens being introduced, causing a lot of damage either directly to us or to our agricultural systems. So I think it's really important to be aware that pathogens just aren't the human pathogens that we might be hearing about in the news. They're everywhere and they're major players. And I think a lot of people, rightly so, have their minds jump to human health immediately when they think about pathogens and the threat posed to society by pathogens. But I think we should really sort of broaden our perspective and understand that it's really important to study pathogens in natural systems and in agriculture, and just across the spectrum of systems to really get a better understanding of how they work, how they evolve, how they spread, and how we can use that knowledge to make the world a safer place. So I'd encourage everyone to just, keep that in mind.
Great. Thank you very much, Ian, and thank you everyone for attending our lab chat today. I hope you all have a great day.
Bye.